In this video, I'm going to give you an introduction to neurogenesis, at least just the basics, so that we can talk about what adult neurogenesis is. So this is a basic overview of neurogenesis and adult neurogenesis. We need to talk about what neurogenesis is, of course. So neurogenesis is the process by which neurons, which I would argue are the most important cell type in the brain. Those of my colleagues that work with astrocytes might disagree. I wouldn't say that astrocytes aren't important. Of course they are, but let's face it. Neurons do the workhorse type work of the brain and make it the most interesting part. Neurogenesis is the process by which neurons are produced by neural stem cells. So this is a diagram. I took this off of Wikipedia, a common core sharing source, illustrating the procedure and the process by which neurogenesis occurs. So early on in development, the brain, or at least the neural tube, is filled with neuroepithelial progenitor cells. Sometimes these cells are also referred to as like radial glial cells or neural progenitor cells. So they'll have typically like a long process that'll extend up to the surface of the brain, and an apical process that'll be nearly adjacent to the ventricle inside the brain, and of course the cell body. Now, one thing that happens is that there's a number of stages that occur during neurogenesis, okay? So, and I like to I break this down into 4 fundamental steps. So neurogenesis really can be broken down into these 4 steps. So the first, of course, is proliferation. So this is cell division. And this doesn't mean that it only occurs once. And in fact, during the process of neurogenesis, especially during development, there will be multiple divisions that will occur. So this regional progenitor cell, there'll be some differentiation that may have occurred, but there'll be this progenitor cell that will divide and create a daughter cell. If that cell is an asynchronous division, an asymmetric division, meaning that like one cell is now going to be a bit different than the other cell that it divided from, that cell is going to change a bit. And can continue to divide as well. So we'll have these initial progenitors, and then there can be intermediate progenitors that occur. So there'll be multiple divisions happening, but in general, you're going to have these divisions happening. So proliferation is generally considered to be the first step. Now, there's a second step that is also happening alongside the proliferation process, can be occurring around the same time. That's why I kind of illustrated it twice. That's called differentiation. So these cells are changing. They're changing the way they look. They're also changing the kinds of proteins that they're expressing. Of course, that means there's changes in the kinds of genes that are being expressed. So there's changes that are occurring, okay? That change is making the cell type change. So that it's going to be a different cell type. It'll have certain features that are just going to be different. It'll be expressing different proteins. We refer to that as differentiation. Now, the other thing you might have noticed is that, we're kind of getting a different position of where these cells are located. And this is true, especially in the developing brain. There'll be a slight difference in positioning of where these cells are. So this would be really close, adjacent to the ventricle of the brain. And then there'll be these cells that will also be dividing that are a little bit more inside. So this is known as the subventricular zone, where these cells will be dividing. And what this means is that some of these cells are actually moving in the brain. They're moving a little bit initially, and that movement we refer to as cell migration. So that is really the third step. But eventually, differentiation will have proceeded so far that there's no longer any more division that's happening, so there's no longer any more proliferation. Differentiation is continuing to happen, but now the cell may be migrating. And so we refer to these cells as being postmitotic, so they no longer can divide anymore. There's no more division that will occur, and then they'll start to be expressing certain features that make them look neuron-like, but they're not entirely neuron-ish. And so these cells can actually migrate long distances within the brain. So that's generally why I kind of put migration at the third position in this list, but it is true that number 2 and number 3 can often occur at the same time. Now the last step, and this is definitely going to be the last step, is integration or incorporation. So differentiation will continue to happen, the cell will get to where it needs to be, and then it'll be integrated into a neural circuit. So this cell will now then grow dendrites and axons and connect and wire up in appropriate ways, hopefully. So that's basically neurogenesis. 4 different steps, breaking it down. But now we're going to talk about neurogenesis as it occurs during development. So before we go on, let's talk a little bit about what neurogenesis looks like in the human brain over time. Most of the neurons that are born in the brain are born by the time an individual is 18 months old. Most of them are actually born before the person is even born. So most of those neurons that are in your brain at this moment, you've actually had them since you were 18 months old, a little toddler. So this is data illustrating this. So it comes from this really excellent paper. I encourage you to take a look at this paper if you want to learn more about migrating neurons in the human brain and how they decline during infancy. This is an immunostaining labeling technique that allows us to see young baby neurons. This is a stain for what's called DCX. We'll talk about that near the end of this presentation. But basically, it's a way of seeing young baby neurons. So the red cells here are the baby neurons. This is the ventricle here. And this is a section that had been taken from some kind of infant brain of a human being. And here's the age of individuals over time. And as you can see, the number of DCX positive neurons, these baby neurons, are relatively high at 1 week, 2 weeks of age, 1 month, 2 months. But by the time an individual is around 9 months or 12 months or even 18 months, it's almost zero. It's not exactly zero, but it's almost zero. So basically what this means is that nearly every single neuron that is born in the brain is born before you're 18 months of age. But there are exceptions, as we're going to learn. So let's talk a little bit about neurogenesis during development. So this is an illustration of what neurogenesis looks like over the course of development. As I mentioned, so basically, the way this is illustrated, this is a complicated looking figure, but it's actually a really nice figure. And it basically is illustrating what I started off with, illustrating the different kinds of cells as there's proliferation, differentiation, migration, and then incorporation. So I'm going to use this to reiterate that development. So early on in development, that's what's on the left-hand side. So this is basically time on the x-axis. So early on in development, we have the ventricular zone. This would be the ventricle here. This would be the outside of the brain, or in this case, maybe even just the neural tube. There are these neuroprogenitor cells, NPCs, that are dividing and giving rise to additional neuroprogenitor cells. So this is known as the progenitor expansion phase. So we're increasing all of our neuroprogenitor cells. Eventually, we start to get differentiation occurring. So we're no longer having symmetrical division happening. Eventually, we start to have asymmetrical division. And there are some neurons that become intermediate progenitor cells, some of the daughter cells end up being intermediate progenitor cells, which eventually turn into neurons. And so you can see that this process of asymmetrical division, so proliferation, there's migration into the subventricular zone. These intermediate progenitor cells can also divide and give rise to daughter cells that will end up being baby neurons. And so these baby neurons might be DCX positive at this stage. You can see that they're really not very neuron looking like, and in fact they kind of look like a squished cell. And they can grow up and kind of crawl along the process of these radioglial cells. So they will migrate up these radioglial cells. So the radioglial cells can serve as kind of a highway system for getting the neurons to their proper place. Eventually, these neurons can continue to differentiate and turn into proper neurons where they will be then integrated. So to reiterate, the 4 steps: proliferation, migration, here, there's differentiation happening as well, and then we have incorporation, when the new baby neuron sends out dendrites and axons to connect to other areas of the brain. Now, one thing about cortical neurogenesis is that the cortex is grown from the inside out. So when we look at the picture of what the adult brain looks like in the cortex, we've got 6 layers, right? We've got the first layer. So here's the pia. Here's the ventricle in here. It'd be way down here, actually, typically. But here's the pia mater, which is the sub portion of the meninges, the dura mater being above that. The first layer is generally free of a lot of cells, especially neurons. Then we've got that combination layer of 2, 3. Then we have layer 4, layer 5, and layer 6. During development, we've got the ventricular zone with all of those neuroprogenitor cells, giving rise to intermediate progenitors. We've got the subcortical plate, which is what SP stands for here. And then 6, 5, 4, and then 3, 2. The brain grows from the inside out. So this is true with the cell division that occurs at the ventricular zone. And then over time, the first layer to get populated of the cortex is the sixth layer. So these neurons will migrate and then create the sixth layer. This now creates scaffolding for then, over time, eventually the cells getting incorporated into the fifth layer. Then over time, we then add the fourth layer, and then the second and third layers, and then the final layer, which isn't being illustrated here. So the brain grows from the inside out, and neurogenesis allows for these neurons to be built up in layers, with the sixth layer being built first, then the fifth, the fourth, and then the second and third. So there's a very famous scientist known as the godfather of neuroscience. His name is Santiago Ramon y Cajal. Here's a picture of him. He's quite pimping in his jacket. I think he's pretty sly. I've seen pictures of him. He looks awesome. Here he is in front of a microscope. He's most famous for some of his developments on improving optics for microscopy, especially for some of his staining techniques, and for describing how the nervous system is structured. One of the things he's really famous for is his extraordinary drawings. So here's some drawings that Santiago Ramon y Cajal had made showing different layers of the cortex. And one thing that I like about this illustration is that, well, we have a couple of different staining methods here. So this is one staining method versus this. But the other thing is that you can get an appreciation for how varied the cortex might be for the different layers. So not all the layers are exactly the same thickness. So here's, layer 2, 3 here, and then this is layer 2, 3 here. And then here's layer 4, and layer 4 is right here, and so on and so forth. And you can see that it can vary quite a bit from different areas of the brain within the same brain. And then, depending upon different stains, you might see other features of the brain. So if you're using a stain that's labeling, say, a lot of the dendrites coming off of neurons, and I believe that this is probably a drawing made from Golgi staining, showing like what neuron architecture looks like in a subset of cells. There's an awful lot of detail that you can get from that. And Santiago Ramon y Cajal used all these techniques to describe the organization of the brain, determining that it was indeed organized as individual cells and not as a combined network of cells, like cells being combined into a single supercells, which is known as a syncytium. But one other thing that Santiago Ramon y Cajal had said is this. So here's a quote. Once the development was ended, and the founts of growth and regeneration of the axons and dendrites dried up irrevocably. In the adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated. It is for the science of the future to change, if possible, this harsh decree. Man, knew how to write back then. Quite beautiful. But what he's basically saying is that there's a lot of change that happens in young brains. Young brains grow, and they change an awful lot during that growth process. And Santiago Ramon y Cajal described a lot of that. But what he's saying is that as far as he could tell, in the adult brain, there is very little change that occurs. Another very famous scientist, Giulio Bizzozero, he was the great-grandfather of neuroscience, you could argue. So he was the guy who trained Camillo Golgi in his work. He had said that the tissues of the human body are labile, stabile, and perennial, so he's talking about the body, but that the nervous system is composed of lifelong lasting cells lacking replicative potentiality. So there is no division that happens in the brain. They're not entirely wrong. It seems like that is generally the case, but there are some important exceptions to that. And that's what we're going to be talking about for the rest of this presentation. So in the 1960s, a professor named Joseph Altman, I believe he was at Purdue at this time, he did some experiments that indicated that there may be new neurons that are formed in the brains of adult mammals. So this is the abstract of his paper. This was published in the journal Science, which is a very high-tier journal. And the title of it is, Are New Neurons Formed in the Brains of Adult Mammals? ? And here's the abstract. In the autoradiographic investigation, the production of brain lesions in rats was combined with intracranial injection of tritiated thymidine. I'll talk about more of what that is in the next slide. Nuclei of numerous glia cells were found labeled in brain regions associated with the traumatized areas. In addition, some neurons and neuroblasts showed labeling, suggesting the possibility of proliferation of neurons in adult rats. This is the very first report of any indication that there's neurogenesis that happens in the brains of adult animals. So let's talk a little bit about what Altman did in this experiment. So this is an experiment that he did in 1962. And what he used is something called tritiated thymidine. Now, we all know that DNA is made of the A, G, C's, and T's. The T is thymidine. And you can make radioactive thymidine if you make it with something that is called tritium. So tritium is a heavy hydrogen. So hydrogen can be radioactive. You can get it out of certain radioactive processes. And if you take that heavy hydrogen, you can make it radioactive. And because it's radioactive, it's going to decompose, like the way radioactivity does, and into simpler elements. And when it does that, it's going to release light. So if you take some tritiated thymidine and you inject it into an animal, any cell that is dividing inside that animal will incorporate some of the tritiated thymidine in newly made DNA. So when a cell is dividing, you're not going to get that happening. And I'll talk a little bit more about this in the next slide on what this looks like. Basically what he did is he did electrolytic lesions of the LGN, the lateral geniculate nucleus. So that's shown here. This is a critical part of the visual system. So this receives input from the contralateral eye and the ipsilateral eye. It actually gets input from both. But then there's a radiation from the LGN to the primary visual cortex. So it's a critical node within the visual pathway. Anyway, so he did this, and then he looked at different brain regions to see if he could see any neurogenesis occurring. Okay, so talking about tritiated thymidine, let's talk a little bit more about this process. This is an illustration of the cell cycle, right? So there's a number of phases. One important phase is the DNA synthesis phase. So when a cell is undergoing division, if it's proliferating, there's going to be new DNA that is made. Every chromosome is duplicated, right? And then we go to the G2 phase, and then we go to the M phase, or the mitosis phase, when the cells get split. So with tritiated thymidine, if you inject an animal with tritiated thymidine, it substitutes for that newly made thymidine in newly made DNA, right? So tritiated thymidine will substitute for regular thymidine in that newly made DNA. The tritiated thymidine that you inject, if it gets stuck into the DNA, it's going to stay there for as long as the cell is there. But any tritiated thymidine that does not get incorporated into DNA is eventually just sort of flushed from the animal's body. And then, so really, you're only labeling cells for about 6 to 12 hours after an injection with tritiated thymidine. And so you will label that, and then that cell is now going to be radioactive because it has radioactive T's in it, which means every daughter cell is also going to have radioactive T's. So at this phase, it would also have radioactive thymidine. And then as it splits, that means that these cells will also have radioactive thymidine. So let's talk a little bit about how you would identify that. Back in the day, before digital cameras, we had like regular cameras that used film. Some of this might be, ancient history to those of you people out there who were born after the year 2000. But believe it or not, we didn't always have digital cameras. We had to use things that had film in them. And the way that film worked is that there were these like silver grains that were built into the film. And if they got hit with anything like light or maybe high fast-moving electrons or photons, what would happen is then these little silver crystals would then change. They would be changed by the light that's hitting them. And so they undergo this sort of chemical change because of the light. And then you undergo this process of developing. So you have to develop the film and then fix the silver grains. So you develop and then fix. And so this is what a negative would be. So if you ever seen like a negative of an image, basically what it looks like is that all those dark grains, the darkness of it, is where there was actually a lot of light. And that's basically how you can make a black and white photo. So this is technology that's been around since the 1800s. The thing is, that's the way, of course, film is, but you can also get that same thing. We call that photographic emulsion. And what you can do is take a slide and dip it in photographic emulsion, so all of these silver grains. So if you have a slide with a bunch of brain slices on it, and if there are any cells in there that have some radioactivity in it, the silver grains just above that radioactive cell will be changed. They're going to be changed chemically. And if you leave it long enough, the tritiated thymidine is going to sort of break down. It's going to release light, right? Because part of what it's doing is releasing radiation. And it's going to activate those silver grains just over the nucleus of that cell. And here are examples from one of Altman's studies showing autoradiographic investigation of cell proliferation in the brains of rats and cats. So he followed up with another study showing the same thing that he had showed in 1962 of examples that look like neurons. Now, he didn't know for sure that they're neurons because at that time we had just basic staining procedures. Morphologically, they look like neurons, but you couldn't say for sure they look like neurons. And he especially saw a lot of these cells in two places, in the hippocampus, and he also saw a whole bunch in the olfactory bulb. Okay, so that's where it was left off with as far as Joseph Altman is concerned. Basically, there was no real further investigation into this phenomenon until the early 80s. So now I'm going to have to talk to you about songbirds. So this is a schematic of the song control system. Later on in this video series, we're going to talk more about the song control system in quite a bit of detail because there's enormous plasticity that occurs in the adult brains of songbirds. But I need to introduce the song control system just a little bit, okay? So I'm just going to talk about the song control system here. So the song control system, this is a cross-section of, a diagram of a cross-section of a bird brain. So this would be the cerebellum, this would be the front of the brain here. This would be then leading to the spinal cord, okay? So this is the telencephalon in a songbird. Then here's the midbrain structures, and then this is the hindbrain structures. All songbird brains are not organized like the way they are in mammals. So the telencephalon in songbirds is not layered like it is in the mammalian cortex. Instead, they have things that are called nuclei. We refer to them as nuclei, but really they're collections of cells, okay? So there's a bunch of neurons that you're going to find within HVC, for instance, and they will send projections. That's what we're illustrating with these arrows. So these would be axons that would synapse onto, say, nucleus RA, or there'd be some other neurons in HVC that would synapse onto nucleus area X, okay, which is part of the basal ganglia. All right, that's as much as I'm going to talk about the song control system per se, except to say that all of these brain areas, we know that they are specifically involved in the production and memorization of song. So I also have another video on my channel describing song control system in my Introduction to Neuroscience lectures. If you want to learn more about just the way the song control system works, feel free to look at that video. For now, all I'm going to focus on is the fact that HVC was heavily investigated, thoroughly investigated by Goldman and Notenbaum in 1983. They basically repeated what Joseph Altman did in the 60s using songbirds this time. Here's a picture of what that looks like. So this is the ventricle here, okay? So they had turned the ventricle on this side. In the ventricle, they could see cells that had a lot of tritiated thymidine. So these would be the neuroprogenitor cells. Let me back up a little bit. Okay, so what Goldman and Notenbaum did is that they injected canaries with tritiated thymidine, and then I think they waited a month, I'm pretty sure. So they waited a whole month, and then they killed them, and then processed their brains for doing the photographic emulsion technique so that they could then see the silver grains overlying the cell. Now, one thing I failed to mention about the silver grain part is that this is a kind of a tedious procedure. It takes about 8 weeks or even longer for the photographic emulsion to lay down enough silver grains for you to see that, yes, okay, this is a radioactive cell. And it's just a very tedious process. You have to do it in a dark room. So if you get any light in the room while you're doing this, you're going to ruin your entire experiment. It's a very sensitive, difficult technique to do. There's better techniques, which is what we're going to talk about here pretty soon. But anyway, so what they did, so Stephen Goldman and Fernando Notenbaum working on this, they had injected birds with treated thymidine, killed them 28 days or maybe a month later, and then did the photographic emulsion technique. So this is the ventricle, and HVC is right below the ventricle. So these would be cells that were within HVC, and they have arrows pointing to cells that have silver grains over the nucleus, indicating that these cells were born the month prior when these animals were injected with treated thymidine. And then these cells are still there because these would be like the other cells that had divided and are still neuroprogenitor cells. Like those cells don't disappear, but then the daughter cell might migrate, differentiate, and then get fully incorporated into a circuit. These cells have an appearance of looking like neurons, but we don't know for sure because, right, they're just basically using the same techniques that Joseph Altman used in the 1960s. So we don't know for sure that these are neurons. How can we show for sure that they're neurons? Well, Fernando Notenbaum followed up with another experiment. He did this with Patton. So this was published in Science in 1984. Okay, and what Patton did, along with Notenbaum, is that they injected animals with treated thymidine. They waited a month, and then they rapidly killed those animals. And instead of slicing the brains and then looking for treated thymidine right away, because they killed them so quickly, they took the brains out of those birds and then did recordings of neurons in HVC. So you can record from cells in order to see if they are neurons. They're going to have electrophysiological properties. They're going to look like neurons. You know, they'll be like action potentials or minis, those kinds of things. When you record from a single neuron, you can also then take that cell and fill it. You can do a filling procedure where you fill it with biotin, and then you can stain the section to fill the whole cell so that you can see that it has a neuron architecture. So this is an example of like what a neuron might look like in HVC. So this would be the entirety of HVC. This is a single neuron that Patton recorded from and then also filled. Okay, so now we can identify neurons, but then we also need to cross-correlate this with the treated thymidine. So the other thing that Patton did is that, so once he did the recordings, then he took those slices and he sliced them thinly again, and then did the photographic emulsion, and then looked to see any cell that he recorded from that looked like a neuron, could he find any of them that also had treated thymidine? And he was able to find 10 cells that he had electrophysiological recordings of, so he could show that they were neurons. The shape was like a neuron. You can see that this is a cell that has like dendrites coming off of it. Same with this one here. And then if you just change the focus a little bit on the microscope, so you go, the silver grains are going to be sitting on top of the tissue, so you can see that the cell is kind of out of focus down below. And so then he changes the focus so that you can see the silver grains, and you can see that this is a cell that's loaded with silver grains, meaning that this was a cell that was born 28 days prior. So he was able to identify newly divided cells that were born only a month before, and that they had neuron-like features, and they were neurons. This was the first demonstration of this, of actual adult neurogenesis. So this was actually like a really big study back in 1984. One thing about this, kind of a historical side note, is that when Patton was doing these experiments, he had to record from over 1,000 neurons. Now, in any one day, if you have a good day of recording and you're keeping track of all these slices, you might have like 6 or 7 neurons. That would be a good day. Any electrophysiologist will tell you, like, there's lots of days where you don't record from any neurons at all. So you would have 0. You would have nothing for those days. And that's just how it goes. So Patton had to do this for years and years and years. And out of recording over 1,000 neurons, he was only able to find 10 that were neurons and happened to be born during that like 6-hour window when these animals were injected with treated thymidine. So he got very lucky, but this kind of drove him insane, and he decided he didn't want to be a part of science after this, from what I understand. Okay, so those are some of the older techniques on historical methods to use to identify adult neurogenesis. had been shown in birds, and then there had been some other data indicating that this also seems to happen in mammals, specifically in the hippocampus and in the olfactory bulb. There are better ways of doing this. So using basic immunohistochemistry techniques is the best way to do this. Any given cell is going to have proteins. There's going to be all kinds of different kinds of proteins that are going to be within that cell. Not every cell is going to have the same kinds of proteins. So like neurons are going to express certain neuron-specific proteins that you're not going to find in, say, glial cells or liver cells or something else. There are some proteins that are expressed in both neurons and glia, but you can find neuron-specific proteins, okay? And then you can do immunostaining for this. So basically, you wash on fixed tissue a primary antibody that's going to be specific to a certain kind of protein, say like a neuron-specific protein, and then you use a secondary antibody that is then going to be a way to label that primary antibody. Now, the secondary antibody is usually going to have some sort of chemical attached to it, some sort of molecule attached to it. Many times we use fluorescence, which I'm going to explain in a second, but there are sometimes techniques that we use, which is using a technique known as horseradish peroxidase. That's what HRPO is. This is like, it's got a peroxidase attached to it, so it's got like an oxide kind of chemistry that is somewhat reactive, and you can do a reaction once you've done the immunostaining to create a colored product. So you'll use diaminobenzidine, which is what DAB is. You do this process, and then you can end up with this color precipitate, which is going to label the cell like an actual color that you can see with your eyes. And then you can use light microscopy to look at the cells and just look at it. So this is different from fluorescence. You can look at it with your eyes or put it under a microscope and look a little bit more closely. So that's known as indirect immunohistochemistry. There's also something known as immunofluorescence. So this might be a technique that some of you are more familiar with, especially if you took say a lab course where you did some immunofluorescence, because this is a more modern technique. It's essentially the same thing. You still have the same primary antibody that you might use to label this protein. So let's say this is a neuron, and this is a neuron-specific protein. An antibody against that neuron-specific protein is going to bind to it. Then you have a secondary antibody that is going to bind to that primary antibody. But instead of using horseradish peroxidase, here we have a fluorescent tag. So this is a fluorescent molecule. When you shine in light, it will then emit light, and that's the signal. So this cell will now glow because it is labeled with this fluorescent tag. So I told you about the tritiated thymidine technique. That isn't a commonly used technique anymore. It was widely used for a couple of decades. Now what we use typically, and what you will see in a lot of the more recent papers on neurogenesis, is using BRDU to label newly divided neurons. So BRDU stands for bromodioxyuridine. Uridine, or uracil, is the opposite of thymidine. So I don't mean opposite, it's the RNA version of thymidine. So, as we know in DNA, we have A, G, C's, and T's. In RNA, we have A, G, C, and U. If you put enough bromodioxyuridine into an animal, some of that will get substituted in newly made DNA where the T's should be. This means, and so this is an example of what this looks like right here. So there's a little bromine group with a couple of other things in there. This is what incorporated BRDU looks like in the DNA. And this is going to make the DNA just slightly unique. It'll be a little bit different. That bromine group is not supposed to be in there, but when you put the synthesized version in there, now it's a little bit different, which is going to make the newly made DNA unique, which means it can be labeled with an antibody. So basically, as I said, BRD substitutes for thymidine in newly made DNA. When we wash on antibodies, we can label cells as they are dividing, like when they're synthesizing newly made DNA, or just after that phase. We can also label cells after the postmitotic phase. And so this is the thing. So just like tritiated thymidine, you can inject an animal with BRDU. You can look 2 hours later to look at actual proliferation. You can maybe wait a day, and then you'll see maybe differentiation occurring or some of the early stages of differentiation and migration. But if you wait a week or a month or a year, you can identify those new neurons from the day that they were born. There are other ways of identifying new cells in the brain, just using basic antibody staining. As we've talked about, as a cell undergoes differentiation, different proteins get expressed. So on the early stages of neurogenesis, you're going to have markers that are going to be more indicative of glia. Many of the neuroprogenitor cells are a glial type cell, and they'll be expressing GFAP. So we can identify those neuroprogenitor cells using things like GFAP and other things like nestin and SOX2. So the combination of these two factors would be indicative of a cell that is a neuroprogenitor cell, especially if it's in the right area of the brain, say in the ventricular zone or the subventricular zone. But after a cell divides and it starts to migrate away, it's going to express things like DCX, Ki67, there's a few others. These are proteins that are going to be expressed in relatively young baby cells, baby neurons. So DCX is going to be indicative of cell that is post-mitotic but isn't fully incorporated into a neural circuit. That's generally what we're going to see with DCX. As a cell matures and as it becomes incorporated, then it's going to express certain things like calbindin or NuN, and this is going to be the most common neuron-specific stain that we're going to see. And so then that will then mean that this is a neuron that is definitely a neuron, right? If it's expressing NuN, it is an incorporated neuron, generally relatively mature. So you're never going to see cells that express NuN and GFAP, because GFAP is going to be something that's going to be specific to glia, and NuN is a way of labeling neurons. So if you were to put GFAP in one antibody channel and NuN in another antibody channel in the same slice, you would see non-overlapping cells. DCX, on the other hand, may have a little bit of NuN in it. It might not. It also may have a little bit of SOX2, but it might not. It probably won't have any GFAP because GFAP is definitely a glial marker, whereas SOX2 tends to linger for a little bit longer. So anyway, you can basically imagine a gradient of proteins that are being expressed as a cell is undergoing differentiation in order to become a neuron, and we know what these proteins are, so you can use this to label them. As I mentioned before, and so what we started off this lecture with, is that we can use antibody staining, such as for double cortin, DCX, to label young neurons. And that's exactly what this figure is showing, right? So this is the ventricular zone right here. So we're revisiting this thing that we talked about at the beginning of this video. This is the ventricle right here. This is the ventricular zone, and just adjacent to the ventricular zone, these are the baby neurons that are migrating and moving their way up into the higher order areas of the brain. So they're migrating away from where they were born, and now they're DCX positive. So the red is the DCX positive cells, and then you can go through brain sections and count the number of DCX positive cells. And again, to reiterate, basically what we see in most areas of the brain, very little neurogenesis occurring. But that isn't true in all areas of the brain. It turns out that there is adult neurogenesis that occurs in the hippocampus, and there's also some adult neurogenesis that occurs in the lateral ventricle. This isn't necessarily true in human beings as far as the lateral ventricle is concerned. I think the evidence is very strong that there's hippocampal neurogenesis, although there's a little bit of debate about it, which is I'm going to talk about in the next video. In addition, there's adult neurogenesis that occurs in the olfactory bulb. This is, and so I'm talking about just mammals here, okay? So I already showed you evidence that adult neurogenesis occurs in birds, it occurs in reptiles and amphibians and in fish. So they generally have a lot of adult neurogenesis that occurs, more so than what we see in mammals. In mammals, we don't see adult neurogenesis occurring in areas like the cortex or the cerebellum or brainstem areas, but in the hippocampus and in the lateral ventricle leading to migrating away to the olfactory bulb, we certainly see adult neurogenesis occurring. This phenomenon of olfactory bulb neurogenesis seems to be largely not a phenomenon that we see in primates. Primates tend to not have very big olfactory bulbs, so it's not surprising to find out that as adults they don't actually have neurogenesis happening into those areas. But many other mammalian species, in particular rodents, which are the most well studied, there's substantial neurogenesis that occurs. So over the next 2 videos, we're going to be talking about adult neurogenesis. The first video is going to be about adult neurogenesis in the hippocampus, and in the second one, we're going to be talking about adult neurogenesis in the olfactory bulb. So if you like this kind of content, please like and subscribe, and I will check you later.